Thank you very much for the invitation to speak here, and I'd like to thank the organising committee for organising this event. So, as Finbar has said, I'm going to talk about the evaluation of novel metal-based drugs for treating drug-resistant microbes. I'm a microbiologist, and I deal with bacteria and fungi, and the vast majority of bacteria and fungi are good. A small percentage cause disease, and an even smaller percentage kill people. We can treat these bacteria because we have antibiotics and cure people. That's a very simple equation. However, because of the development of drug resistance, that equation is no longer working. Bacteria are developing weapons to withstand our drugs. And as a consequence of this, many of the frontline antibiotics are no longer useful. This graph is a famous graph in microbiology because it shows the spread of antibiotic resistance in clinically relevant bacteria. And it follows the development of resistance to a group of drugs known as the fluoroquinolines. They were introduced in 2000. By 2012, 25% of bacterial species were resistant. So in 12 years, 1 in 4 were resistant. Now, 10 years after that, over 80% are resistant, meaning that drug is no longer effective. What's the consequence of this? Well, globally, each year, approximately 10 million people die of bacterial and fungal infections. The vast majority of these people die in the developing world, in Africa and in Asia, but in Europe, around 400,000 die. Many of these infections are untreatable, so the figure is somewhere between 3 and 6 million are resistant to infections. They can't be treated, so the patient is unsavable. Think about it. We're at the end of the COVID-19 pandemic. In the 3 years of the pandemic, 6.5 million people died of COVID. In the same time frame, up to 18 million people died of drug-resistant bacterial infections. So this is a major challenge. Once the pandemic is out of the way, this challenge is getting bigger each year. So what are we able to do about it? Well, we need to develop new ways to treat bacterial infections. And we have 2 strategies funded by the SSPC. One is to repurpose existing compounds that are used, maybe used in a different application, but they're non-toxic. And part of our work has looked at the possibility of using gallium to treat Pseudomonas aeruginosa, a resistant bacterial pathogen. And we've also now worked recently in a project with Darren Griffith and Anne-Marie Healy on using gallium complexes to treat Aspergillus infection, possibly pulmonary infections. The other possibility then is to design novel antimicrobial agents. And the antimicrobial activity of metals has been known for hundreds of years. We can take some of these metals, complex them with other ligands, and change some of their properties to make them more effective for clinical use. Rob has already spoken about the work that we've been involved in looking at the squaramides, and we've also been engaged in work looking at silver complexes with Matthias Tack in UCD. I'm going to talk about one aspect of this work where we use a silver complex to treat the yeast Candida parapsilosis. So this is a fungal pathogen. And it's an emerging fungal pathogen. It wasn't a problem 10 years ago, 20 years ago. It is now. It's capable of colonizing mucosal surfaces in the body, particularly in immunocompromised individuals, so transplant patients, cancer patients, and premature infants. The problem with it is it's resistant to the conventional antifungal agents. We can't treat it. So what are we going to do? Well, we know it's developing an emerging problem. We need to find a weapon against it. And we've evaluated one such weapon. And this is the chemical name of the compound. It's a carbene silver complex. I'll refer to it as SbC3. And here's the silver atom in the center here. And it's complex to this group, which allows the silver to get into the yeast, into the fungal cell better. It increases its ability to cross the membrane. So the first thing that we can do is we can evaluate, does it work against this yeast? And we can carry out an assay where we expose the yeast to different concentrations of the agent, and we see it inhibits growth. That's good. At least we can stop the fungus growing. We mightn't be able to kill it, but we can stop it growing. Then we want to figure out, how does this compound work? And to do this, we can use label-free quantitative proteomics. And in some ways, this is like a mirror. We want to see, how does the compound work on the cell? If we look at the changes in the proteome and see how the proteome changes, it gives us an idea how the compound is working. So to do this, we take our cells, we expose them to our compound, we leave them grow for a number of hours, we extract the proteins from the disrupted cells, we digest the proteins with trypsin, we purify them, we run them on a mass spec, which identifies the proteins and tells us how much is there relative to a control, and then we do data analysis, which I'll show you in a moment. So proteomics is a very powerful technique. Some people say, you're looking at the proteome, why don't you look at the genome? Surely the DNA is where it's at. Well, it's like the plans for a house. An architect draws the plans, that's the information, the DNA, but the builder may change a few things when they're building the house. So the finished product may not represent the plan, the map. It's the same inside the cell. The DNA may not have the full story. So we look at the proteome, and we can see cells exposed to the compound have a dramatic change in their proteome. This gives us this PCA analysis. And by doing this, then, we can begin to analyze what processes are altered or changed in this heat map. So this is the control proteins. These represent the proteins from 2 treatments with our compound. And we can see, for example, that a number of proteins here, the green, for example, are decreased in abundance. So these are involved in biological processes, cellular components, and molecular functions, various functions. And a number are increased, including those associated with oxidative stress. This data then allows us to begin to chart how the cell is responding. And this analysis, we can see various processes such as ribosome biogenesis is decreased in abundance. We can see that respiration is decreased in abundance. Proteins associated with transcription, proteins associated with the synthesis of the cell membrane are decreased in abundance. So the cell is responding to the compound by down-regulating certain activities. One of these is respiration. And using this particular assay, we can see as soon as cells are exposed to the compound, there is a statistically significant decrease in respiration. The cell is stopping respiring. It's also reducing its ability to stick. So this is the process of adherence. So the cell, the yeast cell, can stick to epithelial cells from the patient and grow and infect from there. However, when the cells are exposed to SbC3, they lose this ability. They're no longer pathogenic. Equally, they no longer can produce a biofilm, and a biofilm is essential for their ability to colonize the patient. And the final thing that we can identify is a change in their growth morphology. So these are the control cells, and you can see these hyphal forms, and here they are at a higher magnification. These are like the roots of a tree that help it to allow it to grow into the soil. Same with the fungus, they allow it to dig into tissue. However, when cells are exposed to SBC3, we can see there is none of these hyphal forms. So the cell's ability to infect, the cell's ability to cause disease is reduced. So here's a compound that uses silver in a novel way. The cells respond by increasing their stress response to try and protect themselves. They decrease their ribosomal activity, they decrease respiration, and they decrease their virulence. So now we begin to see we have a weapon. This yeast, Candida parapsilosis, doesn't respond to the conventional antifungal drugs because it's resistant. But it does respond to this. And this compound works in a completely different way to the conventional antifungal drugs. So it gives us an insight into how a new family, a new group of antifungals may be developed for future therapy. So, thank you very much.